So we're going to look at the role of T lymphocytes in the immune system. And just to start off, all you have to do is come up with the names of the 6 types of pathogens, and as an extra question, explain what the role of the lysosomes are in the process of phagocytosis. So if you've already gone through the presentation on phagocytosis, you should be familiar with this image. If you haven't, I'd suggest you go back over that now, find that presentation and watch it, because the whole process of phagocytosis happens before we get to T cells and the actions of T cells. So if you're not familiar with phagocytosis, don't go any further in this one. Go and watch that one and then come back to this. But there are 6 types of pathogen. We've got our viruses, we've got our fungi, we have our protists, because remember they have a nucleus, they are eukaryotic. We have our bacteria, which are prokaryotic, our worms, and our prions. And the extension question was, what is the role of lysosomes in a phagocytosis? Well, the role of lysosomes in phagocytosis is to produce lysozyme enzyme and results in hydrolysis of a pathogen. So if you've had a look at one of the other A-level presentations, either the B cell one or the phagocytosis one, you'll be familiar with this. This is just a brief overview of the entire process, starting with the pathogen, non-specific immunity in terms of barriers, physical barriers, phagocytosis as a process resulting in an antigen presenting cell, all the way through to the T helper cell activation, the roles of T helper cells, and of course the roles of the B cells in terms of antibody production and agglutination. We have already mentioned the terms non-specific and specific immunity. The non-specific immunity is a static response that is common to all pathogens, whereas specific immunity is a response that is targeted to an individual pathogen. They are very specific in shapes and complementary in terms of antibody-antigen interactions, and that's where we get our Bs and T cells. So you should be aware at this point that you should not be using the term white blood cell at A level, and instead should be using terms T lymphocyte and B lymphocyte. You can of course shorten those to T cells and B cells. You should be aware of where T cells that they mature, so they're already there, they just simply mature in the thymus, and the same B cells, it refers to the bone marrow, and you should be aware of the term cell-mediated immunity and humoral immunity, so that whenever they appear in a question, you know specifically the question is linked to T cells or B cells. In order to start the T cells, or specifically the T helper cells, it needs to have an antigen to activate it. The antigens can come from several places. The 4 main ones you need to be aware of are, if you go back over the process of phagocytosis, you should know that the top left picture is a phagocyte that has antigens from a pathogen on its surface, also known as an antigen presenting cell. We can also have cells that have been entered in the body and belong to another organism, for example an organ donation, hence me, myself and Irene. You also have pathogens that will infect the host cell. When they infect the host cell, they will inject their DNA into the host DNA, and the host will essentially start to produce the proteins through transcription and translation to build more virus. Eventually the cell will rupture and the cells will release large amounts of viruses for infection. What can happen is that as part of that process, because the cell is producing large amounts of antigens for the pathogen, some of those antigens are presented on the surface of the cell. So it's another way that T cells can recognize the pathogen based on its antigens. And of course we have ordinary self cells which for some reason have become abnormal or not normal self cells, and they become non-self cells often as a result of mutations in the form of cancer cells. This activity is simply a case of going through each of the statements and putting them into order to describe the stages involved in the cell-mediated immune response. And we're talking about the activation of T helper cells all the way through to the production of large number T helper cells, and then we're going to look specifically next at the 4 rules that have been given. So pause the video for a moment and see if you can work out what the best sequence for those statements is to describe the process. So the last part of the process was to identify the fact that T cells have 4 main functions, and the pictures in front of you are clues as to those 4 specific functions for the T cell clones, T helper cell clones. What I'd like you to do again, pause the video, see if you can work out what they are, and then replay the video to reveal the answers. So pause the video now. Well, the answers are, we've got memory cells, we've got a faster rate of phagocytosis, so they stimulate the phagocytes. They are also responsible for stimulation of B cells to divide, again mitosis, and secrete antibodies, and we also stimulate what are called cytotoxic T cells, or again TC cells. You need to be aware of how TC cells actually work. So at this point, pause the video, look at the 3 pictures, try and work out what the role of cytotoxic T cells is, and then when you're ready, push play and we'll have a little explanation. So pause the video now. Cytotoxic T cells produce a chemical called perforin, and the chemical perforin specifically results in holes or pores developing in the cell surface membrane. It makes it fully permeable or freely permeable, and as a consequence we result in cell death because the cell can no longer control what moves in and out of the cell, which is the primary function of the cell surface membrane. And as a consequence we end up with cell death. So any of the host cells that have been injected with a virus and are producing large amounts of virus or parts of a virus inside the cell, this is how they are destroyed. In this presentation there are a large number of words involved in the process, and there's no alternative but you just have to learn those words off by heart. But what I've got is just a couple of questions to help you skim back over the stuff that's been covered in the presentation so far. So pause the video, go through all the questions, and then when you're ready, if you push play, then you get the answers to all the questions. And just to finish off, what you need to do is you need to make a list of all the keywords and terms from the lesson, the most important words or key terms that you get in the exam, and try and identify and narrow it down to the 7 most important. So of all the key terms and keywords that you can identify, try and narrow it down to just 7 things that are most critical to the concepts that were covered in this lesson on T cells.